So what I thought I'd do today is, we have an amazing assembly of presentations today. And in the next 15 minutes, I'll try to just sort of provide a vignette in alignment with what Carl sort of defined as this multimodal landscape of opportunity and how we're beginning to try and build the required infrastructure that, despite Qinglao's pessimism on behalf of the capacity of clinicians and others to engage these types of efforts, I think it's up to us to build the required tools and resources that bring diversity of expertise together on behalf of the mission. We're not there yet, and I recognize that. But I hope you'll see that there's opportunities to start making progress in that way. And I hope that as you listen to the rest of the talks today, you see reflected in those talks the opportunities we're trying to engage. As Carl mentioned, as you heard yesterday, we've spent a large amount of time and effort to collect large-scale resources, biospecimens, and clinical data on behalf of advancing the mission of discovery. And then these resources are now at the point in time through the combination of philanthropy and NIH have become large-scale resources available for query and discovery. And so we've made this path and continue to do this from the collection of highly curated, high-quality annotated samples that then can be transformed into large-scale data sets. These large-scale data sets then have to become information or essentially interpreted layers of information. And you'll hear about that from the presenters today, looking at each layer of data and even the same data, but looking at it in different ways in order to create new knowledge sets that ultimately drive impact for patients. And you'll see all these boxes that are put around this workflow that really highlight some tenets of opportunity and the way we think about this workflow as a resource. And at the bottom, you'll also see this language around platforms emerging. And these platforms, from our perspective, and many of you have heard me highlight this in the past, are poised to drive accelerated discovery in a way that I think Ken is alluding to. The platforms change the way we do research because they support this non-local, cloud-based infrastructure that allows the global ecosystem to engage these large-scale data sets. They maximize the number of users who can access that data at any point in time, but again, encourage diversity of those users in ways that we hope additional tools and resources expand. And then hopefully connect across those users, even if they don't live in the same city or work in the same hospitals, in order to provide greater access and flow. Part of the reason I was excited Ken was willing to present is because one of the gaps that we constantly face at the academic and healthcare landscape is that it's very natural, if you were to walk around CHOP or any institutions, to ask someone and say, do you as an institution believe in excellence of clinical care? The answer would be yes. Do you as an institution believe in excellence in research? The answer would be yes. Do you as an institution believe in excellence of engineering? There would be a pause, right? There would be a pause around this idea that in order to drive the excellence in research and discovery, you actually have to pair it with excellence of infrastructure and engineering and tools that are not native to the hospital ecosystem. And that's one of the strategies of the community and the opportunity for engagement that I hope we take on as we move forward, and what's required in many ways to advance the field. Is we're very comfortable to engage a pharma company to help us make new drugs. We need to have that same comfort to engage an AWS or the Google or other entities to drive that accelerated discovery because we can't do that alone. Luckily for us, the NIH, despite the failings that Qinglao had also highlighted, has also recognized it, that even the federal ecosystem understands that despite its investment in academic research, they also have to partner with commercial enterprise to build new infrastructure. So leveraging this resource of biosamples and clinical data In 2018, we had the privilege of partnering with NIH to develop the Kids First Data Resource Center, which many of you have heard. And I'll use this as a starting point for that vignette that I spoke about. But in order to make that infrastructure support the Open PBTA, it's really a context of engineering. We had to develop platforms that support the bioinformatics capacity for analyzing data. And you'll hear from people like Arash and others today, and I'll show you a vignette of that, to crunch through large-scale data in a computable environment that's not native to their institution, and it supports doing that data analysis in the cloud. You have to create portals that allow clinicians and other users to at least navigate that platform in ways that allow them to understand what data or what subjects are available. And you do have to create knowledge bases that distill that large-scale data into meaningful clinically relevant information in ways that retain security, governance, access, and supports further collaborations between both NIH and institutional and consortia-based levels. So on top of all that infrastructure, you have to have that layer of security, governance, and frameworks that provide the landscape for that. And as you'll hear today, each of these samples are where we look at DNA, RNA, or protein or metabolites. Each one of those layers is further divided into these complex set of files that describe a layer of that data that has a meaning to biology, and in many ways require specific expertise to analyze the DNA sequence or the RNA sequence, the expression analysis, the immune microenvironment as defined by expression analysis, the ecDNA that can be extracted from DNA sequences. All of that has to happen through individual laboratories looking at the data and then reassembled to create a picture for the patient and the discovery setting. We'll see if I'm successful in going through some of that framework for you. And again, I'll hop around because of time, but just to highlight both the opportunity and the challenges we'll face to support this diverse community around these resources. So here I just logged in to the Kids First platform, where essentially all CBTN data resides. And you can see that here, right? So we have 6,100 subjects within the platform within CBTN. And you can see as a table view, a listing of those subjects and histologies and some basic information. But you can also see that for each subject, how many files are available for that. And I'll show you in a second that information. But overall, across these 6,100 participants, there are more than 45,000 files generated. So that means for 45,000 files for that many participants, those files represent each layer of data, right? And to be transparent, I don't know what each one of those files means, as an individual. And there are very few people here who would be able to understand and interpret all 45,000 files for those individuals. We have investigators who focus on specific layers through histology. So here I'm clicking on a high-grade glioma subset of those files. But even within high-grade gliomas, there are more than 6,000 files just for those 500 participants that have already been analyzed. And then, as Carl mentioned, even if you just look within any particular context, you'll see that there are a variety of different clinical and other available data modalities. So you'll see that in addition to the sequencing read, the pathology or histology slides, we'll also see radiology and other resources for those. And let me just click on one of these so you can review them. So the first goal for us to engage community is to provide access to source, right? To provide access to source data, because we understand that that source data can be interpreted differentially by different resources. And you'll see some examples of that. I see Lucas in the audience, and he'll present later. We were sequencing and analyzing genomic data, from pediatric brain tumors for some period of time, missing, right, ecDNA signal in that data until Lucas came along. But the cool thing that Lucas did when he came along is he said, well, I want to analyze it, but I want everybody else to analyze it. And I want everybody else to analyze it from the get-go. And so working with him, we actually incorporated that workflow, you'll see it here, into the Cavatica workflow. So now every new genome that comes through our system is automatically reanalyzed with new knowledge derived by a community stakeholder who was initially not funded by NIH, but by a foundation to support their discovery process. Within that setting, I just want to highlight that it can get even more impressive and complex. So here I just chose one subject, this one single patient. And this one participant already has 168 files associated with them. But the dedication and effort of teams to continuously add resources to this one subject is further highlighted when I'm just going to dig in to all the resources that are available for that subject. So this one subject has all of these biospecimens attached to that subject. And if I look at the derived data that it's potentially more amenable to the clinical interface, you'll see here that we can immediately see all of the mutations characterized, not just at one sample, but across 4 different events. And in fact, those 4 different events are not just surgical samples, but if I look at any one of them, you'll see some information about those events. And importantly, you'll notice that some of those events are actually cell lines and derived renewable data sets from that cohort. You can see this a little bit better, I think, in this view here. So this is a new view that's not yet available within the CBio portal instance, but it highlights our continued effort to represent information to different stakeholders. So when I, as a biologist, look at a patient, I largely care about these mutational settings and there's then changes in those DNA and RNA layers. But a clinician actually cares about what treatment they received and when they received it, right? Their course of treatment, the specific events of progression that occurred across that timeline, and when and how they received this combination of treatments and events all the way from the start of diagnosis, and unfortunately for this particular patient, till the end of their care. And then Carl highlighted our efforts to expand the imaging data set. So here you can see a pre-op image and a post-op image. And I can click on this particular image and come immediately into the framework where all the imaging data is available for that particular subject. And I'm not a radiologist, but I can click on buttons. And so I'll click on one button and you'll see that even I went all the way right from source data to analyze results to the clinical care workflow for that subject, what they received, the imaging that they received, the actual MRIs for that subject, that now a second set of informaticians, typically completely different than those that care about DNA and RNA, can start selecting which images they want to process, run through their AI algorithms, and then link to the other algorithms that are analyzing the genomic. But all of that has to happen, right, across distinct groups, right, with very different expertise, right, but they have to be brought together into an ecosystem or framework that they can communicate between each other, compare their results, and then continue to layer data on this. All of this data for this subject was not generated at 1 week or 1 month. It was generated asynchronously across programs and resources. This particular subject that has both their samples analyzed, their cell lines analyzed, is also part of the proteomic projects that you'll hear about today. The same subject now also has proteins and phosphoprotein data attached to it, information that the genomicists can't themselves understand. So it really takes a community to understand each one of these layers. And we'll get better and better at providing this data to the right people at the right time. But those people have to be part of that same ecosystem in order to drive that knowledge creation process on behalf of both the discovery And our ultimate hope is that all of this can happen in near real time on behalf of the individual patient who needs the care informed by these data. And that's the challenge we hope to address, and we can only do it through this amalgamation of what I call a motley crew of the dedicated humans on behalf of pediatric cancer and making a difference. So I appreciate your, as Carl said, partnership in this experiment to do something completely different in ways that I think will change and transform the landscape. Thanks so much.